Welcome to the Explainer. When we think about the global fight against malaria, our minds usually go straight to bed nets and bug spray, right? But there is actually a silent, incredibly high-stakes battle happening right now at the microscopic level. We are literally in an evolutionary arms race between our best diagnostic tools and a highly evasive, shape-shifting parasite. And this isn't just about laboratory data. This microscopic arms race dictates whether millions of people get the life-saving treatment they need. So today, we're going to look at the fascinating and, frankly, kind of alarming ways the malaria parasite is learning to hide from our tests, and exactly how scientists are racing to outsmart it. To really understand this arms race, we first need to look at our primary weapon, rapid diagnostic tests, or RDTs. See, for most of the 20th century, if you wanted to diagnose malaria, you needed a microscope, a highly trained technician, and reliable electricity. Obviously, that's incredibly difficult to maintain in remote, resource-limited settings. So RDTs absolutely revolutionized point-of-care diagnosis. These simple lateral flow tests work a lot like an at-home COVID or pregnancy test. You apply a drop of blood, and within 15 to 30 minutes, without needing a single watt of electricity, you get a parasite-based diagnosis. It was a massive, game-changing victory for global health.
But as you'll see, this victory is currently under threat. Here's our agenda for today's Explainer. 1. The rise of malaria RDTs. 2. The HRP2 problem. 3. Limitations of PLDH tests. 4. Hunting for new biomarkers. And finally, 5. Next-generation diagnostic tools.
Okay, let's jump right into Section 1, the rise of malaria RDTs.
To see how this whole dilemma builds, we have to jump back to 1991. That's when the scientific inertia around malaria diagnostics was completely broken. Researchers characterized a specific biomarker called histidine-rich protein 2, or HRP2 for short. Now, HRP2 is a protein excreted in massive amounts by Plasmodium falciparum, which is the deadliest species of the malaria parasite. It's heat-stable and highly specific to this exact parasite. Because the parasite turns out so much of this protein throughout its entire life cycle in human blood, it became the absolute gold standard target for modern rapid tests.
Moving to Section 2, the HRP2 problem. So, we found a highly abundant protein and built billions of tests to detect it. Problem solved, right? Well, unfortunately, no. The parasite, it turns out, is an invisible enemy that's actively adapting to evade detection. The widespread use of these HRP2-based tests has revealed a really complicated set of limitations. First, and this is the craziest one, the parasite is literally deleting its own genes. Specifically, the PFHRP2 and PFHRP3 genes. I'll get to that in just a second. Second, we have post-treatment antigen persistence. Because HRP2 clears from the bloodstream so slowly, the antigen can stick around for 4 to 5 weeks after a patient is completely cured. This causes false positives, which totally confuses doctors who might think the treatment failed. Third, there's the prozone effect. This is a bizarre phenomenon where a surprisingly high parasite density actually overwhelms the test and causes a false negative. It's kind of like a crowd so densely packed that nobody can actually move to get through the door. And finally, the antibodies on the test strips themselves are vulnerable to heat degradation in tropical climates. But I really want to focus on that first point, the gene deletions, because this brilliantly illustrates the sheer urgency of the problem. 17%. In a study conducted in Nigeria, an astonishing 17% of analyzed Plasmodium isolates had simply deleted the PFHRP2 gene. Lab experiments have actually shown that the parasite doesn't really need HRP2 to survive and grow. So under the intense selective pressure of our diagnostic tests, it pulled off a magic trick. It evolved to drop the gene entirely. That makes it completely invisible to our best, most widely used tests. It's literally evolution happening right in front of us.
Up next, Section 3, limitations of PLDH tests.
If the parasite is successfully hiding from our best tests, the logical next question is, why not just use a different test? Enter our backup biomarker, PLDH, or Plasmodium lactate dehydrogenase. It's an essential enzyme found across all human-infecting malaria species. But when you compare these two primary targets side by side, you see the issue. HRP2 gives us high sensitivity. It's great at catching low-level infections, but it has that slow clearance time causing false positives. PLDH is the exact opposite. It clears super fast, usually within 3 to 5 days after successful treatment, meaning it's excellent for confirming an active infection right now. However, it has significantly lower analytical sensitivity. The data behind this sensitivity gap is pretty stark. To get a reliable positive reading, meaning over 95% of tests turning positive, PLDH requires a concentration of roughly 45 nanograms per milliliter in the blood. HRP2, on the other hand, requires a mere 4 nanograms per milliliter. Notice that massive 10-fold difference? Relying solely on PLDH as our backup means we risk missing a huge number of low-density infections. And worst of all, we could even drive the parasite to mutate this essential enzyme too. Which brings us to Section 4, hunting for new biomarkers. So if HRP2 is being deleted by the parasite and PLDH isn't sensitive enough to catch every case, we're staring down a pretty dangerous diagnostic gap. But researchers didn't just guess what to look for next. They played detective. They adopted a fascinating, two-pronged, systematic approach to interrogate the parasite's biology. Step 1 was a massive systematic literature review, hunting for published evidence on literally any non-traditional malaria biomarkers. Step 2 was an in silico analysis. That means using advanced computer models to systematically scan massive databases of parasite proteomics data, specifically looking for proteins with favorable diagnostic features. Just to give you an idea of the sheer volume of this hunt, the initial search across major medical databases like PubMed and Web of Science identified 3,914 publications. They were casting an incredibly wide net, looking at host biomarkers like cytokines, as well as proteins originating from the parasite itself. But through a really rigorous funneling process, we're talking excluding duplicates, irrelevant studies, and targets that were just way too complex for a simple rapid test, they eventually narrowed that massive field of almost 4,000 studies down to exactly 98 unique biomarker candidates. The goal was incredibly strict. Find single parasite proteins that are easy to detect, don't require heavy medical infrastructure, and can survive the blistering heat of endemic regions. Finally, Section 5, Next-Generation Diagnostic Tools. Out of those 98 candidates, what actually survived that brutal dual validation? Well, this complementary approach led researchers to 2 incredibly promising novel lead candidates, GAPDH and DHFR-TS. Both of these showed really favorable diagnostic properties in the computer models and in the published literature. GAPDH is actually a biomarker proposed for various conditions, but the immediate development need here is validating it for clinical decision-making specifically in malaria. DHFR-TS is also incredibly promising, but its immediate development hurdle is creating high-affinity reagents. Basically, we need to engineer better antibodies to grab onto this specific protein in a rapid test format so we can prove it works reliably in the real world. Now, while we wait for these brand new biomarkers to hit the market, innovation definitely hasn't stopped on our current tests. We're seeing the powerful emergence of ultra-sensitive RDTs, or URDTs. These new tests boast a 10-fold lower limit of detection compared to standard RDTs. In recent studies in high-transmission areas like Mali, these URDTs had a 50% probability of detecting HRP2 at an incredibly low threshold of just 207 picograms per milliliter. To put that in perspective, a picogram is 1/trillionth of a gram. It is essentially looking for a microscopic needle in a bloodstream haystack. So while we figure out how to fully navigate those gene deletions, these ultra-sensitive tests are helping us catch the absolute lowest level infections that standard tests would completely miss. We are literally in a microscopic chess match right now. The parasite deletes genes to become invisible. We respond by hunting through its proteomic data for new targets like GAPDH, and by engineering ultra-sensitive tests that can detect mere picograms of remaining antigens. All of this brings us to a final, pretty provocative thought. Can our diagnostic tools outpace parasite evolution to finally eliminate malaria? The answer relies entirely on continuous innovation, rigorous tracking of these mutations, and understanding the science we've explored today. Thank you so much for joining this explainer, and keep questioning the science around you.